Hello there, I'm Jamain Hatton, and I'm starting a series that will look at genetic inheritance, particularly for my students who are having a difficulty understanding inheritance. So we have to look at some key terms. One of those key terms happen to be the idea of inheritance. What is inheritance, you might ask? Well, inheritance is just simply where genes, characteristics, genetic information are passed from mother and father to offspring, right? So parents transfer their genetic information to their offspring. That is genetic inheritance. And because we have two parents, an individual will often have, always, an individual will always have two sets of genome. Now, what is a genome? The genome is a sum of all the genes that an individual inherits, all the genes in an individual's body. And since you have two parents, you would have two sets of genome, one from your mother, one from father, and then together now, those individual genome will come together to make your individual genome, right? Some basic terms we have to understand before we actually go on to understanding genetics and genetic inheritance. One of those major terms is the term of chromosome. Now, what is a chromosome? It says just here that it is a thread-like structure that is found in the nucleus and is passed from parents to offspring. It carries genetic information in the form of genes, and that's very, very important, because if we are to understand how inheritance works, we have to understand chromosomes. The chromosomes are what basically allows for information to be passed from person to person, parents to offspring. And that information that we are talking about is in the form of genes. So the genes really is a part, one single part within a large chromosome, right? And if you were to remember from previous studies, I guess, from lower forms when you were studying, you actually have 23 chromosomes from both of your parents. 23 from mom, 23 from dad. Together, you have 46 chromosomes, or what we refer to as 23 pairs, right? Each of those pairs, each of those sets, 23, 23, from both of your parents. And all of these chromosomes carry different characteristics on them, right? So what are genes now? Like I've said, genes is a part of a chromosome, and that part basically controls different characteristics such as your height, intelligence, eye color, complexion, etc. Certain characteristics, like for example eye color, multiple genes located at different parts of different chromosomes actually allow for that determination. So some of them are very simple, where you have one single gene on one single part of a chromosome coding for a particular characteristic, and then there are some where you have multiple genes from multiple different chromosomes coding for the same characteristic, right? So that is gene and chromosome. So now we come to allele. An allele is a different form of a gene. An individual will always have two different forms of a gene. And when they inherit those two different forms of that gene, that is said to have, they are said to have two different sets of alleles. And can you understand, or can you tell me why an individual might have two different forms of a gene or two different alleles? What do you think? What might account for two different alleles? Every individual has two different alleles for the same gene. Two different alleles for the same gene. What might account for that? If you said parents, you would indeed be correct. Because an individual has two parents, each of their parents will contribute one allele to the offspring. So one allele from each parent. Right, so let's say the allele we are talking about, and you'll see when we do genetic crossing, the alleles actually are letters, capital letters and common letters. Let's say the allele we are talking about is height, and we will always use the dominant allele. We'll talk about this term in just a bit. Capital T to represent that dominant allele. So let's say your mother gives you that dominant allele and your father gives you that recessive allele, or the smallness. Let's say your mother was short, your father was tall. So your father gives you that tall allele. I think I said the mother just now, but let's say the mother gives you the tall allele and the father gives you the short allele. This, you'll get one from mother, one from father. One from father, one from mother. Right? So an individual always would have two sets of an allele for a specific characteristic. For example, height, because they're getting genes from both mother and father would contribute to the single characteristic of height. Your genotype now is the specific alleles of a gene present in an individual. Take, for instance, we are saying that an individual would have inherited two different sets of allele from both of their parents. In the case I used above just now, the person actually got the allele, the person actually got the tall allele, let's say they got it from their mother, and they got a short allele from their father. This here together, that capital T and that common T, would be their genotype. This is their genotype. This is their specific alleles that they inherited for a particular trait, right? So that's your genotype, or we simply say genetic makeup, right? The phenotype, on the other hand, is not these capital T and common T's, but it's actually what we observe in an individual. So the phenotype are those characteristics that we can see. So what can we see in an individual? We can see that the person is tall. We can see the person is tall, or we can see the person is short. Whatever we see, that is the characteristic that will be classed as phenotype. If we see that the person is intelligent, or we see the person is not so intelligent, that is a characteristic we observe. If someone can taste a particular thing, that would be their phenotype, whether they can taste it or not. So again, the phenotype is that characteristic that is observed in an individual. Right? Let's go this again quickly. Chromosome is the thing that you inherit from parents. You inherit a total of 46. On those chromosomes, we have genes. Those genes code for particular things. Those genes are responsible for characteristics such as height, intelligence, eye color, complexion, every possible thing an individual can examine themselves to have. Alleles. Alleles are a different form or forms of genes that you inherit from both parents, mother and father. Your genotype would be the specific set of characteristics that you observed or you inherited from both parents, right? Your phenotype would be those observed characteristics. So let's look now at some other key terms. There is the word trait. A trait is a physical, specific characteristic, right? So let's say your ability to roll your tongue, whether your earlobe is attached or not, whether your thumb actually has an angle when it's upright. Those are traits. Some traits are dominant while others are recessive. The dominant trait, when it's present, will be shown by a capital letter. Let's say D, capital D. Let's say B, capital B, capital T. It matters not what the characteristic is, but you can choose to represent it with letters. Oftentimes, if we're talking about height, we would say T for tall, right? So that would be your dominant characteristic. So once present, dominant characteristics are always expressed. What does that mean? If your mother was tall and your father was short and they gave you those two genes, once your mother gave you the tall gene, it matters not who gave you. Once you have one copy of that tall allele, that capital T, the child will always be tall. Well, of course, genetics can play a part. When I say genetics, I mean environment can play a part where the individual might actually not be eating enough to actually get tall. But once the individual gets that tall gene, that dominant allele, from either of their parents, the individual has the capability of becoming tall. The recessive allele, for simplicity's sake, I'm saying here that it's the weaker version. It's not necessarily the weaker version, but it just simply means that whenever a recessive allele is present, it is not often expressed, it's often masked, especially when a dominant allele is present. Think, for instance, one parent gave the tall allele, capital T, and the other one gave the short allele, common T. Whenever capital T is present, capital T will always be expressed. But when common T is present, common T is masked. The common T is the recessive allele, right? For you to actually see somebody become short, you actually have to get an inheritance where they actually got two short alleles from both of their parents. So both parents should have given them common T's, and that is a case where they would actually be short as children, right? Then we have two terms, heterozygous and homozygous. And because I want you to understand this, I'm going to slow it down just to ensure that these two terms are understood. So what are heterozygous traits? Heterozygous refers to the inheritance of different forms of particular genes from both parents. So what does that mean? It's the same thing that we have talked about already. So heterozygous, when you think about heterosexual, you're attracted to different persons other than your current sex. So difference is seen there. So one person got the tall allele, that capital T, one person got the short allele. One person gave the tall allele, one person gave the short allele. This is what we call heterozygous. Two different forms of the same gene. Two different forms of the same gene, right? And then homozygous, homo means same, homo, same. So if homo means same, and it's saying here two identical forms of a gene are inherited, right? Two identical forms are inherited. So what does that mean? Small t, or large T, large T. So capital, capital, or common, common, right? But remember, it's homozygous because it's the same allele being present. Common T, capital T, capital T. You think you understand? All right, let me attempt to go this one a little bit over just for clarity. So homozygous simply means the person is getting both of the same alleles. So if the person got a T, a height, and they're tall, both parents gave them that two tall alleles. If the person is short and it's homozygous, two small t's were actually attained. And because we have understood the using of the word dominant and recessive, now we can bring the genetics into it. And the genetics is simple. Remember just now we said the capital T was dominant and the recessive allele was actually the small t, right? So let's erase all of this and go again when it comes down to the bottom here to see if we understand what we have said so far. Let me change my color and let's go. So if heterozygous is two different forms of the allele, we have capital T and we have common T. Homozygous, capital T, capital T. What do we call this? It's homozygous. It will always start with homozygous because we have the same two alleles. But we can go on to describe this a little further to say homozygous dominant. Why? Because it's two of the dominant allele. We can see that there. Two of the dominant allele. Homozygous dominant. Let's say they were two small t's. It will still be homozygous. Why? Because we have two of the same small allele, and it's going to be called recessive afterwards. Homozygous recessive. Two of that weaker version, if I am to say that. But it's not necessary to actually go on to say heterozygous dominant or heterozygous recessive. Why do you think it's not necessary to say that? Because if you notice, heterozygous actually means two different forms of the allele. So you don't need to go on to specify that it was actually heterozygous dominant and heterozygous recessive, because heterozygous mean that they actually got two different forms of the allele. Alright? But you can also go on, but you should go on to say homozygous dominant or homozygous recessive, because the person can actually inherit two opposite things, so you need to specify what is the homozygous you're talking about. Good. So let's take a break now. Go on and drop a comment in the comment section. Let me understand if there are any specific difficulties you might have with this. Now let's put it into practice. So when the father of inheritance, Gregor Mendel, yes, that's his name. Gregor Mendel discovered this inheritance in the 19th century. Little did he know this was actually going to be the greatest invention probably for humankind, because it therefore meant that people can actually control in some way or the other what genes are passed on and what genes are not. All right? So let's see. Gregor did an experiment where he crossed a tall pea plant with a short pea plant. So he had, let's say, the pollen grains from a tall pea plant and the flower of a short pea plant, and it was pollinated, right? So he had all the offsprings becoming tall. And at that point, it was puzzling to him because he thought they would actually become medium-sized, but they actually became tall. And let's understand what Gregor actually discovered when he discovered this. So to do this, I'm going to do what we call a Punnett square. And the Punnett square is just showing us two different sets of characteristics being transferred. Well, not two different sets. Let's go to it again. The same characteristic, but it's two different sets of alleles are being crossed. Remember, this, let me just change my color quickly. This allele here was from one parent, and these alleles here were from another parent. So parent 1, parent 2. All right? Good. So let's go back now to our crossing. So what will happen if we cross these tall pea plants with these short pea plants? Let's see. So capital T times common T will give us what? Capital T, common T. What would we get when we cross capital T and common T again? Same thing. Capital T, common T. And capital T, common T, capital T, common T. And that's what you can see there. Once a dominant allele is present in the crossing, the individual will always see. You will always have all tall offspring. So once a dominant allele is present, the characteristic is always transferred. Let's go this one again, just for clarity. So remember, we are getting two different sets of alleles from both parents.
And this Punnett square is a crossing that's used to actually show the possibility of getting a certain trait from a certain parent, right? So let's see. So we are crossing them. Let's say the first parent was the parent with the tallness, the tall pea plant. And then the other trait is the short pea plant. All right? So let's see. Capital T, capital T, capital T, common T rather. Can you see that? Here, capital T, common T. So that's what we get there. When we cross capital T and a common T, it's the same thing we are doing essentially, if you notice that. So all will have a capital T. And once all of them has the capital T, all of the offspring will actually be tall. So to interpret this, we have to do what we call probabilities or a chance, right? So what does this mean? If you're asked to interpret, we can interpret phenotype and we can interpret genotype, right? Whenever we do a crossing, whatever our offspring are, these here will be termed our F1 generation. Right, so the F1 generation is what we get after we cross a parent, two sets of parents, right? Good. Our genotype is what we have as our alleles within our genes, right? Our phenotype is what we observed. So if we can say that we just had 4, I'm erasing this to get some space because I'm not sure how to create space. I probably can zoom out and create space. Can't I do that? Let me see. I'm still getting to learn the software. I don't think I'm getting to do that. So yes, if we created just now a crossing that led to all the offspring, all 4 of the offspring, actually having a genotype of heterozygous, 1 big T, 1 small t, what would the phenotype be? The phenotype, because they have that tall characteristic, that tall allele present, all of the offspring would be tall. So that's 100% tall, right? And the genotype would be 100% heterozygous. Think you understand that? So 100% tall, because remember, phenotype is what we see, and the genotype is what the genes or the alleles that were present determine. So because we have 1 tall T and 1 short t, or 1 small t, we have 2 different sets of alleles. 1 is dominant, 1 is recessive. When we have that combination is referred to as our heterozygous pair, right? So there we have 100% heterozygous. Now let's work some CXC questions. So this question is saying, Mary and James both carry sickle cell trait. So they both have the sickle cell trait. What is the chance, that's what we were talking about just a bit, probability, same as chance. What is the chance that a child born to them would suffer the sickle cell anemia, Explain your answer using the aid of a genetic diagram, and use the following symbols: A, normal, and S for sickle cell. So, a question like this is worth 6 marks in the exam. And as you can see, it will be 6 very, very simple marks. Let's go. So the first thing we do, let's set up our genetic crossing. Remember, James and Mary are the 2 parents, and they said they both carry the sickle cell trait. If they both carry the sickle cell trait, it therefore meant that none of them were full-blown sicklers. None of them had sickle cell anemia. They just carried that trait. And remember, the trait was S. So both parents had S. So what is the other allele? The normal allele is A. So we put that A there. Both parents were normal because they just carried the trait, right? So let's see what is the possibility of their children suffering from sickle cell anemia. Let's do the crossing. A, what do we have? Same alleles from both parents. What do we call this term here? We call this homozygous because it's the same allele. But what do we say? Homozygous dominant? Yes, homozygous dominant. They got the two normal allele, right? The normal allele will always be more dominant than the recessive allele. Good. So in the next instance, we have A, S. So we have capital A and S. What is this individual here? This will be a carrier. This person is a carrier, similar to the parents. What do we have here? SA, we always put the normal allele first. We always put the dominant allele first, and we put the recessive allele after that. So it will always be the dominant allele first. So even though we have S times A, it's actually going to be written AS. The dominant allele always goes first. And finally, let me put this one here in a color that we can all see and appreciate. S times S, we would get SS. And if they said that the S was actually the sickle cell trait, the sickle cell allele, if you have two copies of that same sickle cell allele, therefore this would be the individual who will suffer from the sickle cell anemia. This individual will be a full-blown sickler, that we would call in the medical terminology. So that doesn't answer the question. This is just part of the question. The question that CXC asked here is to use a genetic diagram to predict the chance that a child born to them would suffer from sickle cell. So you have to go on to say some interpretation now of this. And what they want to know, whether the child will have sickle cell anemia. So we can ignore 1, 2, 3 and just focus on the stuff that they want you to focus on, which is this bit here, right? So there are 4 possibilities, and there is 1 out of 4. That equates to 25%. So therefore, you can surmise that 25% probability, or a chance of 25%, exists that the child will suffer from sickle cell anemia. Right? Let's go there quickly again. Both parents were carrying the trait. So it's capital A, normal allele, and the sickle cell trait, which is that S. Good. And when you cross them, we get 1 homozygous recessive. We have 2 heterozygous. Can you see those 2 heterozygous? Look, they're there. 2 different sets of alleles. Can you see that? 2 different sets of alleles, and that would be the heterozygous. And then we have 1 homozygous dominant here. So when this woman gets pregnant, when Mary gets pregnant, there will always be a 25% chance that one of her child will actually have sickle cell disease. And one of her child, 25% again, will be absolutely normal. But there's a 50% chance, can you see that? There's a 50% chance of her children being carriers. Can you see that? Because it's the AS gene would be that carrier gene. I hope this is making sense. And this is a question I created. So it's saying fetal thiocarbamide, let's just say PTC. PTC is a compound that can be tasted by people who carry the gene with at least one dominant allele, and we're going to call that dominant allele T. So once you have that capital T, that dominant allele, you can taste this compound. And it's not tasted by individuals who are homozygous recessive. So once you have a dominant allele, capital T, you will taste this PTC. If you have a recessive allele, you will not taste this PTC, right? So it says Quincy is a fifth form student and is saddened by the fact that he cannot taste PTC, but both of his parents can. Use your knowledge of inheritance to answer the following question. What is the genotype of Quincy's parents likely to be? So remember, Quincy cannot taste PTC. And if he cannot taste it, it therefore meant that he inherited that allele, or these sets of alleles. So he must have had two recessive alleles for him not to taste or be able to taste PTC. Remember, the question said that if the individual inherits one dominant allele, that is enough to taste PTC. If he cannot taste it, it therefore means that he inherited two recessive alleles. So if Quincy inherited two recessive alleles, but his parents are both able to taste PTC, what does that tell us? Quincy might be a blow child. That's a probability. He could actually have another father. But the probability that exists, really, the strongest probability, is the fact that the parents were actually heterozygous. Both of the parents had that recessive allele. And if we are to cross that these homozygous pairs, we will see what we are talking about. So look at this. Let's change our color. Let's do it now. So what do we have here? Big T, homozygous, dominant. Big T, small t, heterozygous. Small t, big T, small t, heterozygous. Small t, so homozygous, recessive. This here is 1/4. How do we know 4? There's 4 possibilities. 1 here, 2 here, 3 here, 4. So 1 of 4 is homozygous recessive. So this equates to 25%. This 25% is the probability that Quincy actually inherited. That probability is what he inherited. So this is what Quincy is. Quincy is homozygous, recessive. He inherited that trait from those two parents that were carriers of this ability to not taste PTC. Right? So the parents were obviously heterozygous, having two sets of different alleles for the same trait. We have used the Punnett square to show the condition. What is Quincy's genotype? Quincy's genotype is homozygous, recessive. And they're asking you now, finally, if Quincy were to marry Alina, who inherited both normal tasting genes from her parents, what will be the phenotype of their F1 generation? What will be the phenotype as a result of this crossing? So, let's see. Remember, it doesn't matter where you put the parents, right? You can put them anywhere. You can put them on this side here, or you can put them on that top part there. Doesn't matter who goes where. It will still result in the same crossing. So let's say Quincy was... What was his genotype? Yes, he was homozygous recessive. Remember that from the previous slide? Good. And Alina, it says she inherited both normal tasting alleles from her parents. So that means what? She is homozygous dominant. Can you see that? Yes, she's homozygous dominant. And if she is homozygous dominant and her boyfriend or her husband is homozygous recessive, what will we have when we cross them? Look at that. Big T, small t always will give you heterozygous. Good. Big T, small t again, heterozygous. Big T, small t, big T, small t. So we have 100% will actually be heterozygous. What does that mean? If one T is enough, if one big T is enough, one dominant allele is enough to actually taste PTC, all the children they have, all the children, once Quincy is the father, will always be able to taste PTC. Why is that? The dominant allele, once it's present, will always mask that recessive allele from that gene. I hope that was a bit clear. I hope this helps you a little bit. But we have to talk about a little thing before we move on, before we finish today's session. So what I want to talk about is the term diploid and haploid, because you can be asked in an exam to talk about or defining haploid and diploid. So diploid is the 46 chromosomes. Everybody in every cell of their body has 46 chromosomes. Why? Because at the time of fertilization, you inherited 23 from parent 1 and 23 from parent 2. But inside the testis and the ovaries of your parents, or any boy or any girl, there will always be a haploid number. What does that mean? Meiosis occurs, and what meiosis will do is reduce this 46 down to 23. So 23 would be in one sperm, and 23 will be in one oocyte, or ovum, or egg. So 23 will be in the egg, 23 will be in the sperm. When sexual reproduction takes place, the egg will be fertilized with a spermatozoa, right? So when that spermatozoa fertilizes the egg, we would have a restoration of the diploid number. So what does that mean? It means the 23 that one of your parents give will be added to the 23 of the other parent. So together we will have a zygote with 46 chromosomes. And this is what we call 2N, diploid. We have N in one sperm, 23, and N in one egg, 23. So the N is just 23. When we have two sets of the N, when we have two sets of 23 coming together later on, we would have 46, right? So N plus N give you 2N. 23 plus 23 gives you 46. Right? So the diploid is just simply a chromosome with 46, an egg. Well, diploid is a cell with 46 chromosomes, while the haploid is a cell with 23 chromosomes. And if we are to recap, what cell would have 23 chromosomes? Good. 23 chromosomes will only be in our sperm cell and our egg cell. But once fertilization takes place, we'll have a fusion of the sperm and the egg cell, and we will have a restoration or a completeness happening where there will be 23 from parent 1 and 23 from parent 2, two haploid sets going back to give us now what we refer to as the diploid number, which is 2N. So N from here, this is 23, this is 23. These two come together and they will give us 2N, which is 46.
And that concludes my session on genetics.